Oh, welcome back, everybody. We're now in our technical talks session, so we have a handful of presentations here that will be more technical, scientific engineering in nature. And each one of our technical talks will be around 20 minutes. Starting first with talk on fMRI processing pipelines by Micha Burkhardt from University of Oldenburg in Germany. So welcome, Micha.
That's correct. Thank you so much. Hello, everyone. Thanks for having me. Just a little warning for me in Germany, it's now a bit past midnight, and after a long day of work, my brain slowly starts to give up on me, but I hope the talk will still be fine
My
nonetheless. So,
apologies. I'm sure you'll do fine.
nothing to apologize. I've had a blast so far. So, first and foremost, I want to say that I'm very happy to be able to present today, and I'm also really humbled by the previous talks. And even though the topics were not that usual for me, so there will be a slight shift in the topic now. So my name is Micha Burkhardt. I'm a first year PhD student in the Psychological Methods and Statistics Lab at the University of Oldenburg in Germany. And I mostly work in the domain of cognitive neuroscience, but I also have a strong interest in open, robust, and reproducible science. And so I think this is quite fitting in the context of, I think, reproducibility, validity, and integrity, I think, as stated by the conference aims for the technical talks. So my talk today will be a bit different than the keynotes, as I already mentioned. But I hope that some general concepts might still be applicable or interesting for anyone listening. So the title of today's talk, of my talk today, is Quantifying Similarities between fMRI Processing Pipelines for Efficient Multiverse Analysis. And as this is quite a complex title, I think it's best to start with a general problem setup. So we in Oldenburg work a lot with functional MRI data, but fMRI data is inherently noisy. It requires long preprocessing pipelines and also complex statistical analyses. And this is problematic as while creating these pipelines, researchers are often faced with a plethora of arbitrary yet defensible decisions. So these decisions, for example, could include things like how to correct for MRI scanner artifacts or movement correction or how to derive functional networks from the data. And all these decisions also contribute to something which is sometimes referred to as researchers' degrees of freedom, which can introduce bias and also contributes to the replication crisis we in psychology and many other fields of science are currently facing. So if we have this issue, what are potential ways of improving this? So one recently proposed solution is called multiverse analysis. And briefly, this describes running multiple equally defensible analysis pipelines in parallel instead of just the traditional single analysis pipeline, which you often see published. And then multiverse analysis involves to integrating over the results. Excuse me. So you can see an illustration of this in the figure where just like three binary decisions in a multiverse or in analysis pipeline lead to a total of 8 different combinations or what we call universes. And one can then use these results to integrate over them or to further investigate any patterns that emerge, trying to build or formulate better theories and hypotheses for future work. So this aims to help with the issue of flexibility in the data, in data processing pipelines by forcing researchers to think about possible alternative decisions and by having to explicitly state and evaluate them, which then in return reduces bias from selective reporting and improves the robustness and reproducibility of findings. So after having settled that, the next questions regarding the title would be, why do we need multiverse analysis to be more efficient? And I think the answer to this is quite intuitive, pretty simple, as you already probably saw on the last slide. Oops, that was accident. As you probably saw on the last slide, the number of universes in multiverse analysis increases quickly when the space of decisions is large, and such is the case in neuroimaging studies. This means it can quickly become computationally infeasible to run the entire multiverse. So previous literature has already acknowledged this issue. For example, Jessica Davlon and colleagues proposed to subsample the multiverse, meaning that converging on a smaller subset of analysis pipelines that are representative of the variability in the results of the entire multiverse. And to do so, they create a two-dimensional search space from the output similarities for a subset of subjects. Now you can see this at the bottom right, where each point in the plot represents a result of an analysis pipeline, and the proximity in space then represents their similarity. And they then apply a machine learning algorithm to subsample the space, showing that sampling with only 50 points, for example, already closely matches the empirical distribution of the results. So this is an interesting finding. However, it has two key limitations. So first, this empirical space requires one to calculate all analysis pipelines. So this is using a lot of computational resources, also resulting in large carbon footprint. And second, a subset of the subjects is needed to be used for the creation of the search space. So we all know that, especially in like the clinical context, subjects are a very valuable resource. So considering these limitations, we propose that it should be possible to efficiently subsample such a space of decisions just by the analysis pipelines themselves. So while this is an intuitive idea, this is also based on the empirical observation that similar pipelines, so the output of similar pipelines, these are often close to each other in the two-dimensional space. So this means that we aim to assess the similarity between pipelines by creating some kind of lower dimensional embedding. So this does not necessarily need to be two-dimensional, based on the configuration of the pipelines themselves and not on their outputs. So we just want to compare like the structural differences in the pipeline design. So what kind of data do we use for that? So from previous work by Daniel Crisanto and colleagues, we have 220 fMRI analysis pipelines in the context of network neuroscience and brain behavior associations derived from literature. And these pipelines can be seen in the figure below. They contain 61 distinct data processing and analysis steps. So these are the data processing steps are the circles, the nodes, with the connections between them being the number of papers that went from one data processing or analysis step to the next. And an interesting fact is that no two papers applied the same analysis pipeline. So every single pipeline in this review is different. So starting from this resource, we then want to assess the similarity between pipelines. And at the bottom, you see how two pipelines could look. In this case, they're very similar, but two steps of cleaning and brain parcellation are swapped in that case. So how similar or how dissimilar are such pipelines? So for this, we applied three classical measures. I don't want to dive into too much detail here, but namely the Jaccard index, Hamming distance, and Levenshtein distance. And we also introduced a novel method based on a graph convolutional neural network. This is because we realized we can interpret the space of the analysis pipelines shown in a previous slide as a network with the analysis steps being the nodes and the papers going from one step to another step as the edges. And then we apply an algorithm called Deep Graph Infomax to derive some implicit features from these nodes, from these analysis steps, which we then base our own similarity measure on. So I don't want to dive into too much detail here, but essentially we have the set of established similarity measures and then this newly developed approach. And how exactly does this work? So simply put, a graph neural network learns some local or global graph embeddings by aggregating information from nodes and their neighboring nodes. So if this data processing step is used, is it often used with the second step, or does it always come before, come after? So the graph neural network can kind of learn this implicit information about the graph. And then these learned embeddings and it will create some nodal embeddings. And these embeddings can then be used for downstream tasks, like you can see in the green figure, such as graph classification, node classification, and so forth. In our case, then to derive the similarities. And then the Deep Graph Infomax algorithm, which we use, you can see illustrated at the bottom right. It's an extension to graph convolutional networks, or to the graph convolutional network in our case. And it is an unsupervised algorithm that is trained to best distinguish between real versus corrupted sections of the graph. And importantly, it uses not only local features of the graph, meaning which information is connected with which step, but we can also capture more global structure of this graph by adding additional features, such as the frequency of an analysis step being used in literature, its in degree, out degree, what kind of like group of processing it belongs to, like structural processing, functional preprocessing, and so forth. And what are the results? So when we apply these measures, we first see, or you can see in the matrices at the top, that there's a reasonable overlap between all these measures, with correlations between 0.15, so pretty low, up to 0.75, pretty high for the different similarity measures. And when we look at the ARI, which is the adjusted rand index as a measure of cluster overlap, we see similar results. But we can also see that three methods stand out and show a higher overlap with each other. This is the three ones in the center. And interestingly, as you can see in the bottom figure, these three methods are the similarity measures that consider the ordering information within the pipeline, which tells us that there seems to be some important information there. So this bottom figure, as you see, it lists all the measures we applied, with the first ones being based, or the first two being based on the embeddings in our graph neural network, and the last three being the traditional methods. And this general overlap between methods tells us that our new approach seems to work as intended, and is a promising avenue for further work on top of it. And a second finding is that we see the emergence of two distinct patterns. So we see that different measures handle length difference in pipelines quite differently. So if we have two pipelines that are highly similar, but just vary in their length, meaning that like one analysis pipeline contains some additional steps, maybe in the end or somewhere in between, our graph neural network approach will consider them to be fairly dissimilar, while the traditional measures, such as Levenshtein and Hamming distance, view them as to be fairly similar. And the second pattern emerges in cases where two broad processing categories of data cleaning and data parcellation are switched. So the graph neural network still considers these pipelines to be fairly similar, while, for example, Levenshtein distance consistently treats them as dissimilar. So there's seems to be constant pattern there. So as a summary, the graph convolutional network seems to be promising for capturing relevant information about the similarity between pipelines. And the different measures we evaluated show a general overlap between them, but also display distinct behaviors and patterns. The advantage of our newly proposed approach for creating such a search space for an efficient subsampling of the multiverse would thus lie in not requiring us to calculate the entire multiverse and not to use valuable subjects for creating a search space. I would also like to emphasize that this approach is method agnostic, which means it could be applied to other areas of research and data analysis as well. However, future work is needed as we need to test the performance or accuracy of this newly developed method and also the other methods on empirical data. So this will already be done in a limited scope in the revision of our conference paper. However, doing this exhaustively will be a big effort, and we first need to be more certain about the validity of this approach. And if we find that the search space derived by any of these measures to be satisfactory, we then need to integrate them into this entire multiverse workflow, where we aim to apply the machine learning approaches for the subsampling procedure. So to wrap it up, we hope that this approach will go on to become a small part in making the entire process of data analysis and publication a bit more robust and contribute to improved reproducibility of future findings. And with that being said, I already arrived at the end of my talk. I'm happy to take any questions now if there's time. But if you have further feedback, questions, ideas, feel free to reach out to either Daniel or me. Thank you so much again for having me.
Yes, I have a question, but let me first throw it out to anybody else who might have a burning question. If not,
Okay.
then, yeah,
Sorry.
please, Adam. Yeah, go ahead. Go
All
ahead.
right. So, how do you decide when two steps in a pipe, two possible steps in a pipeline are the same step versus different steps? So, for example, you could, on the one hand, you could be running the exact same software with the same settings, like the same parameters, or there are cases where, or you might run the same software with slightly different parameters, or you might even run two implementations that are in some sense doing the same thing, but with a somewhat different implementation. Maybe one of them makes some approximations, the other doesn't. Like we recently encountered this with fMRI processing with the NODDI feature extraction. Someone we were working with had a MATLAB library and a Python library that supposedly did the same thing, but one was a lot sort of less precise about it and ran orders of magnitude faster. So at what point do you say a process, two things are the same thing?
Yeah, that's a great question and also a tough one. So in this concept of multiverse analysis, often we use the term equally defensible. And equally defensible, it's something that's up to be argued with also. But I think the first good thing it does, it requires us as researchers to actively think about our decisions and why we take them, and also about alternatives. So in this case, equally defensible, for example, would mean there's entire papers out on these categories. So what is equally defensible, what is not equally defensible, but it means basically that you have no solid idea of why this one MATLAB toolbox, for example, should be methodologically superior to the other before actually running it. And if there's no like clear indication on why one decision should be superior compared to the other, then it would always make sense to include such a decision in a multiverse. I don't know if this answers your questions or makes sense. Um,
Oh yeah,
yeah. So it's still up to
yeah,
the researcher. It's not an completely objective decision, right? It's always, it stays subjective. It just
okay.
yeah.
Though just as part and parcel of that, though, you still want to communicate what your pipelines are in a way that's mutually intelligible between different researchers. And so it would might be worthwhile to investigate like standardized ways of, well, I don't think there's a universally sort of accepted standard with these things, but just like it's worth looking into different sort of formats for representing research pipelines rather than just picking some arbitrary sort of coding strategy for late coming up with label names for different steps you identify in your literature review.
Well, maybe some of you folks can get together and create a consensus group that develops such a standard. I don't want to fall too behind schedule, but I did want to make a shout out for placing this kind of work in the original experimental context. So, what was the original experimental paradigm for the fMRI studies that were investigated here? I mean, there are all kinds of different experimental paradigms for fMRI studies done with human subjects, and so one of my important questions would be: do you then subject your interpretations to the quote unquote smell test, where you place it back into the context of what was the original framework of the original experimental paradigm and application? I mean, did this involve fMRI studies that were simple vision hearing stimuli response observations? Were there fMRI studies that looked at completely different kinds of issues like um, memory, cognition, emotion? So I think the point that I'm making is that I think this kind of work needs to be placed in the framework and context of the original experimental paradigm. You need to close the loop and then subject it to the common sense and intuition tests on whether or not the pipeline that you selected as either better or worse yields results for the actual experimental question that was posed by the original functional MRI paradigm that was investigated. So I'll leave it there. Please consider that for your discussion in your manuscript. It's a very, very important question, and it's the one of you know connecting these kinds of data analytics to the underlying human behavior, the underlying human biology, and the real world validation of whether or not you're getting results that make sense or are just gobbledygook. So we'll leave it there. Let's try and stay on schedule.